Hello everyone. We have learned hypoxia. Today we will do experiment different types of hypoxia. Now first, let's review hypoxia. Definition of hypoxia. Classification of hypoxia. You think, what is hypoxia? Hypoxia can be defined as a deficiency in neither the delivery or the utilization of oxygen at a tissue level, which can be lead to change in function, metabolism, and even structure of the body. Now, according to the etiology and parameters of oxygen, hypoxia is divided into 4 types: hypotonic hypoxia, chemic hypoxia, circulatory hypoxia, and exogenous hypoxia. Now today, we will copy 3 types hypoxia. Section 1. Experiment objective. Now it includes 3. Now first, copy 3 types of hypoxia models in mice. The second, observe the change of respiration, color of skin in different types of hypoxia. Now the last, master the treatment for the different hypoxia. Section 2. Experiment animals. 7 mature mice. Only 1 newborn mouse. Section 3. Equipments and drugs. The next, I will introduce. Now, different types of hypoxia. Now, it is drugs for hypotonic hypoxia. Carbon monoxide poisoning, and treatment. Injections. Now the big injection contained oxygen. The small contained carbon monoxide. The drug is sodium nitrate. Now it is sodium sulfate. It is methylene blue. Now it is normoxylane. Now it is potassium cyanide. Now it is mature mice. It is newborn mice. You must observe respiratory frequency and depth. Observe swell time. Observe the color of skin, mucosa, and the liver. Now section 3. Mice. Now today we will copy 3 types hypoxia: hypotonic hypoxia, chemic hypoxia. It includes carbon monoxide poisoning, sodium nitrate poisoning, and the last, ketogenous hypoxia, potassium cyanide poisoning. Now the first, hypotonic hypoxia. Now the first type hypoxia, hypotonic hypoxia. Now let's review. What is hypotonic hypoxia? Hypoxic hypoxia is characterized by the decrease of partial pressure of oxygen in artery It's also called hypotonic hypoxia. Characterized partial pressure of oxygen in artery decrease. Now, partial pressure of oxygen in artery, the normal value is 100 millimeters mercury. If partial pressure of oxygen in artery is 60 to 100 millimeters mercury, pulmonary ventilation is normal. So supply oxygen is normal, no hypoxia. If partial pressure of oxygen in artery is less than 60, so lead to saturation of oxygen decrease, oxygen content decrease, hemoglobin combined oxygen decrease, so supply oxygen decrease and happen hypotonic hypoxia. What can lead to hypotonic hypoxia? Etiology of hypotonic hypoxia include 3. Now first, decrease partial pressure of oxygen of inspired air decrease. So it will lead to partial pressure of oxygen in alveoli decrease and lead to partial pressure of oxygen in artery decrease. So happen hypotonic hypoxia. The second, external respiratory dysfunction. So lead to artery partial pressure of oxygen in artery decrease. Also happen hypotonic hypoxia. The last, venous to artery shot. Venous blood content oxygen is less than arterial blood. So if venous to artery shot, so lead to arterial blood content oxygen decrease and lead to partial pressure of oxygen in artery blood decrease. So supply oxygen decrease and happen hypotonic hypoxia. Now it is etiology of hypotonic hypoxia. Now do experiment. Now the first, put soda lime into a jar. The next, put a mature mouse into the jar. Now you observe the respiratory rate every 10 seconds. Now you observe the color of skin and the lip. Now it is normal respiration and the color. The next, put newborn mouse packed with air net into the jar. Observe the color of skin and the lip. The next, cover the jar. So the animals inspired oxygen will decrease. And the next, lead to partial pressure of oxygen in artery blood decrease. So happen hypotonic hypoxia. Now the next, you observe the respiratory rate every 3 minutes, the color of skin, and the swallow time. Now newborn mouse, you only observe swallow time. Now you think. Now respiration, increase or decrease? What color of lip and skin? Now which animal swallow longer? Mature mouse or newborn mouse? Announcements. Observe the fluctuation of abdomen. Keep a quiet environment to decrease errors of the frequency of respiration. Now, put soda lime into a jar. You think, why What is function? Now put a mature mouse into the jar. The next, pack the newborn mouse. Don't damage your hands. Also, put the newborn mouse into the jar. Now you observe the normal respiration and the normal color. Now cover the jar, so the animal inspired oxygen will decrease, so the animals happen hypotonic hypoxia. Now you observe the respiration and the color of skin. You find respiration first increase and the next decrease. Color of skin is bluish. After about half an hour, the mature mouse died. Now you think the newborn mouse die or survive? Newborn mouse didn't die. It survived. It is survived. Why it survived? Now results the experiment. The normal respiration mature mouse is 29. Color of skin is light pink. Now during the hypotonic hypoxia, alteration of respiration. Respiration is 37, 39, 45, 40, now 28, 20, 17, 12, and stop. Now color of skin and lip is bluish. Now color is bluish. What happened? Cyanosis. You think, what is cyanosis? Now survival time about 25 minutes. Newborn mouse now survive, didn't die. Analysis and summary. For question, what's the action of the soda lime? The second, which one survive longer? Mature or newborn mouse? Why? The third, what change of the respiration of hypotonic hypoxia? Why? Hypotonic hypoxia leads to cyanosis. Color of skin is bluish. Why hypotonic hypoxia leads to cyanosis? Now you think. You can answer or no. Now the first, what's the action of the soda lime? Soda lime is composed of calcium oxide and sodium hydroxide, which can absorb the carbon dioxide and decrease the area of the experiment. Now the second question. Newborn mouse survive longer than mature, which may be due to the following reason. The second, newborn mouse had a very low rate of the metabolism, so it needs fewer oxygen. The second, hemoglobin of the newborn is more than that of the mature mouse. The third, energy is supplied by glycolysis in newborn mouse, while mature mouse depend on aerobic oxidation. So newborn mouse survive longer. Now, how did the respiratory change of the hypotonic hypoxia? Why? When pulse pressure is 30 to 100 mm mercury, there is almost no change in volume of pulmonary ventilation. Now, if pulse pressure of oxygen is less than 60, now so the low pulse pressure of oxygen can stimulate the carotid body and the aortic body. So the next will stimulate a respiratory center, so lead to respiration increase and hypopnea. So during the hypotonic hypoxia, the mouse we observed pulse respiration first increase. Why the next respiration decrease? Because the hypoxia more, so pulse pressure of oxygen in artery decrease more. If the parameter is less than 30, can inhibit respiratory center, so lead to respiration decrease. So during the hypotonic hypoxia, pulse pressure now respiration first increase and the next decrease. Now respiration increase because pulse pressure of oxygen is less than 60. Now you think the other types hypoxia, for example, chemic hypoxia, circulatory hypoxia, and ketogenous hypoxia, have or no have respiration increase. Now because the three types of hypoxia, pulse pressure of oxygen in artery is normal, so no hypopnea. Understand? Now color of skin and mucous membrane. In our blood, hemoglobin combined oxygen is called oxyhemoglobin. Now don't combine the oxygen, the hemoglobin called deoxyhemoglobin. Now in normal blood, deoxyhemoglobin is less than 2.6 grams in 100 millimeters blood. Now if oxyhemoglobin decrease and lead to deoxyhemoglobin increase, if deoxyhemoglobin more than 5 grams in 100 millimeters blood, now color of skin, lip, and nail are bluish. It's called cyanosis. Now cyanosis, if the concentration of deoxyhemoglobin in blood in capillaries is more than 5 grams in 100 millimeters blood, the color of skin, nails, lips, and the mucous membrane are bluish. Now the definition you might remember. cyanosis. Now, hypercyanosis, which increase deoxyhemoglobin or oxyhemoglobin, deoxyhemoglobin increase. Now, during the hypotonic hypoxia, what alteration? Deoxyhemoglobin and oxyhemoglobin, which increase, which decrease. Now, during the hypotonic hypoxia, because partial pressure of oxygen in artery less than 60, so lead to oxygen saturation decrease. So hemoglobin combined oxygen decrease and lead to oxyhemoglobin decrease. Now oxygen content in artery decrease. So oxyhemoglobin decrease and deoxyhemoglobin increase and hypercyanosis. Now color of skin, nail, lip are bluish. So we observed the color of the mouth. Color is bluish. Now it is hypotonic hypoxia. Second experiment, chemic hypoxia. Now let's review. Chemic hypoxia refers to the altered affinity of hemoglobin for oxygen or decrease in concentration of hemoglobin in the blood. So during the chemic hypoxia, maybe affinity of hemoglobin for oxygen increase, so lead to oxyhemoglobin liberate oxygen difficult, so lead to oxygen move into the tissue cell decrease. Now the other concentration of hemoglobin decrease, so carried oxygen decrease, supply oxygen decrease, and happen hypoxia. The hypoxia called the chemic hypoxia. Now characteristic during the chemic hypoxia, inspired oxygen normal, external respiration normal, so partial pressure of oxygen in artery is normal. Now, because affinity of hemoglobin for oxygen increase, concentration of hemoglobin decrease, so lead to combined oxygen decrease. Now lead to oxygen content decrease. Etiology and the mechanism of chemic hypoxia. Now etiology include anemia, concentration of hemoglobin decrease, so happen chemic hypoxia, carbon monoxide poisoning, and methemoglobinemia. Affinity of hemoglobin for oxygen increase in the tissue capillary blood. Oxyhemoglobin liberate oxygen difficult, so lead to oxygen move into the tissue cell decrease and happen hypoxia. Now today we will do carbon monoxide poisoning and sodium nitrate poisoning. Now the first, carbon monoxide poisoning. Now the first, mature mouse A and mature mouse B. Now first the mature mouse A, put a mature mouse into drop number 1. Now you observe the respiration and the color of skin and the lip. Now it is normal respiration and the color of skin and the lip. Now the next, cover the jar and inject carbon monoxide about 5 milliliter into the jar. So the animal A will inhale carbon monoxide, so happen carbon monoxide poisoning. Observe the respiratory rate every 3 minutes, the color of lip skin, and observe the swallow time. So the animal A hypochemic hypoxia, carbon monoxide poisoning. Now animal B. Now put another mature mouse into drug number 2. Now observe the respiratory rate, color of skin and lip. Now the same similar to animal A. Now cover the drug and inject carbon monoxide 5 ml into the drug. Now you observe the manifestation of the animal. Now the next, inject oxygen about 50 ml to the drug immediately after the mouse collapse. Now you observe the swallow time. So animal B first inhale carbon monoxide and the next inhale oxygen. Now the animal will die or survive. Now the big injection contain oxygen, the small contain carbon monoxide. Now first, take a mouse into the drug. Now you observe the respiration and the color of skin and the lip. Now it is normal. Now close the jar. Inject carbon monoxide. Regulate it. Now inject about 5 ml carbon monoxide. Now close. Now you observe the manifestation of the carbon monoxide poisoning. Now you observe the color of lip. What color you think? Now, why happened the color? Now respiration increase or decrease. Now during the hypotonic hypoxia, respiration first increase and the next decrease. Now chemic hypoxia, respiration no increase. Now respiration decrease, even stop. Now the animal collapse. Now collapse. Now respiration decrease. You observe the color. What color? Cherry red. Now the mouse die. Now take another, also into the jar. Now you observe the normal respiration and the normal color of skin. Now open. Close. Now the same. Inject about 5 milliliters carbon monoxide. Now inject about 5 milliliters. Now prepare the oxygen. Now big injection contained oxygen, but it closed. So the animal inhale carbon monoxide and hyphen carbon monoxide poisoning. After collapse, inject the oxygen. Now it must open because oxygen more. If it closed, maybe lead to the bottle break. So you must open. Now you observe the manifestation of the animal. Now the animal first inhale carbon monoxide and hyphen carbon monoxide poisoning, and next inhale oxygen. Now treatment. Now you observe the manifestation. The animal will die or survive. Now the animal survive. Don't. Now carbon monoxide poisoning. 2 animals. The first animal inspired the carbon monoxide, inhale carbon monoxide, and animal B first inhale carbon monoxide and the next inhale oxygen. Now the first animal die, the second animal survive. Now results. Animal A, normal respiration 28, color normal, light pink. Now respiration. After inhaling carbon monoxide, respiration 20, 13. Now color, cherry red. Survival time, about 2 minutes, about 3 minutes. Now animal B, normal respiration 30. Now color, light pink. The animal didn't die, survive. Now it is carbon monoxide poisoning. Analysis and summary have 4 questions. First, which type hypoxia that's caused by carbon monoxide poisoning belong to? The second, what's the color of skin and the lip? Why happened the color? Now the third, what's the pathogenesis of carbon monoxide poisoning? Now the last, why can oxygen treat carbon monoxide poisoning. Now, carbon monoxide poisoning, the hypoxia belongs to chemic hypoxia. Now, color of skin and lip is cherry red. Why the color is cherry red? Because in the blood have a lot of carboxyhemoglobin. Why have carboxyhemoglobin? Now it is the pathogenesis of carbon monoxide poisoning. Now, if inhale carbon monoxide, now carbon monoxide can move into the capillary blood. Now hemoglobin can combine with carbon monoxide and form carboxyhemoglobin. Now carboxyhemoglobin can't carry the oxygen, so lead to supply oxygen decrease and hypoxia. Affinity of hemoglobin for carbon monoxide is more than hemoglobin for oxygen. Now carboxyhemoglobin liberates hemoglobin very, very slowly, so also lead to hypoxia. Now the second pathogenesis, carbon monoxide can inhibit glycolysis, so lead to produce 2,3-DPG decrease. Now it decrease, what will happen? Now will lead to dissociation curve, shift left or shift right. Now dissociation curve, shift left. So affinity of hemoglobin for oxygen. Affinity increase or decrease? Now shift left. Affinity increase. Now affinity increase, what will happen? Affinity of hemoglobin for oxygen increase. So in the tissue capillary blood, oxyhemoglobin liberate oxygen will difficult, so lead to oxygen move into the tissue cell decrease and hypoxia. So inhale carbon monoxide, inhale carbon monoxide, so will happen hypoxia. Now the hypoxia belong to chemic hypoxia. Now because in the blood form a lot of carboxyhemoglobin, color of carboxyhemoglobin is cherry red, so color of carbon monoxide poisoning is cherry red. Because carboxyhemoglobin is cherry red. Now the last, why can inhaling oxygen treat carbon monoxide poisoning? Because inhaling oxygen can lead to dissolved oxygen increase, so supply oxygen increase. Now the second, inhaling oxygen can enhance oxyhemoglobin liberate oxygen, so oxygen move into the tissue cell will increase. Now inhaling oxygen can treat carbon monoxide poisoning. Now the next, the chemic hypoxia, sodium nitrate poisoning. Now it also belongs to chemic hypoxia. Now they're similar to carbon monoxide poisoning. Now animal A, you first observe respiration and the color of skin and lip. It's normal respiration and the color of skin. Now the next, inject sodium nitrate about 0.3 milliliter into the abdominal cavity. Now the next, inject normal saline, now into the abdominal cavity. Now the animal A first inject sodium nitrate, and the next inject normal saline. Normal saline have function or no function? No function. Now the animal A hypoxemic hypoxia. Now you observe the respiratory rate every 3 minutes, the color of lip, skin, and you also observe the swallow time. Now you think the animal survive or die. Now animal B. Also, you observe the respiratory rate, color of skin and lip. And next, inject sodium nitrate into the abdominal cavity. Now the next, inject methylene blue into the abdominal cavity. So animal B first inject sodium nitrate, and hypoxemic hypoxia, and the next inject methylene blue. Now methylene blue can treat nitrate poisoning. Why it can treat? Now the next, you observe the survival time. The animal, you think the animal die or survive? The animal maybe die. Now you observe. Now the first animal. Now you inject the sodium nitrate. Now the animal A inject the sodium nitrate. From an abdominal cavity, inject. About 0.3. So the animal hypoxemic hypoxia. Now you observe the respiration and observe the color of lip and the skin. Now the animal first inject sodium nitrate, and the next inject normal saline. Animal B first, now it is methylene blue. Prepare. Now first, the animal B first also inject the sodium nitrate. First inject sodium nitrate, and the next inject the methylene blue. Now you observe the two animal manifestation. Now animal B, respiration decrease and collapse. Now animal B collapse. Now, sorry. Now it is animal A. Animal A first inject the sodium nitrate, and next inject the normal saline. Now hypoxemic chemical hypoxia. Now it is animal B. First inject the sodium nitrate and next inject the methylene blue. Now the animal first happen chemical hypoxia and next treatment. So you observe the manifestation. Animal A, animal B, which will die. Now you observed. Animal A die, animal B now recover. Now results. The third experiment. Animal A and animal B. The mouse A normal respiration 31. Now normal color is light pink. Respiration now 29, 23, and 15. Stop. Now color is coffee. Now survival time about 3 minutes. Animal B. Normal respiration is 29. Color light pink. Now it is recover, survive, didn't die. Now you know. Animal A first inject sodium nitrate and happen chemical hypoxia. Now not treatment. Animal B first inject sodium nitrate and next inject methylene blue. Now the animal first happen chemical hypoxia and next treatment. So the animal recover, survive, didn't die. Analysis and summary. Also have 4 questions. First, which types hypoxia? Now nitrate poisoning. Now what's the color? Now what's the mechanism of nitrate poisoning? Why can methylene blue treat nitrate poisoning? Now the first, nitrate poisoning belongs to chemical hypoxia. What's the color? We know, coffee. Nitrate poisoning pathogenesis. What's the mechanism of nitrate poisoning? Now include 2. The first, hemoglobin can carry the oxygen. Which carry oxygen? Now ferrous state or ferric state. Now ferrous state can carry the oxygen. Now nitrate poisoning, nitrate is oxidizer. So it can oxidize the ferrous state from ferric state and form methemoglobin. Now it called the methemoglobin. Now ferric state can't carry the oxygen, so lead to supply oxygen decrease and happen hypoxia. Now the other, one hemoglobin have 4 ferrous state. If one ferrous state form ferric state, the other ferrous state can carry the oxygen, but affinity of ferrous state for oxygen increase. So during the tissue capillary blood, hemoglobin liberate oxygen will difficult. So lead to oxygen move into the tissue cell decrease and happen hypoxia. Now it is a mechanism of nitrate poisoning. Because during the nitrate poisoning from ferric state, ferric state color is coffee. So the poisoning color of skin is coffee. Why methylene blue can treat the nitrate poisoning? Now, methylene blue is reducer. So lead to ferric state from ferrous state. So it also carried oxygen. So methylene blue can treat nitrate poisoning because methylene blue is reducer. Nitrate is oxidizer. Now the next, ketogenous hypoxia. Now what is ketogenous hypoxia? It refers to the oxygen is supplied adequately to the tissue, while the tissue cell can't make use of the oxygen supplied to them. Now, ketogenous hypoxia supply oxygen in normal, but utilize oxygen decrease. Now, characteristic. During the ketogenous hypoxia, intravenous difference of oxygen content decrease. Now, etiology. The etiology includes three. The first, inhibition of mitochondrial oxidative phosphorylation. The second, mitochondria injured. The last, absence of vitamin. Now, the three utilize oxygen decrease, so produce ATP decrease. Now, potassium cyanide poisoning now belong to ketogenous hypoxia. The cyanide can lead to tissue cell utilize oxygen decrease. Now, mature mouse A. First, you observe the respiratory rate, color of skin and lip. Now, inject cyanide 0.2 in the abdominal cavity. The next, inject normal saline. So the animal happen ketogenous hypoxia because normal saline no function. Observe the respiratory rate every 3 minutes, the color of lips, skin, survival time. Now, mature mouse B. First, you observe the normal respiration and the color of skin and lip. Now, inject cyanide 0.2. Now, the next. Now, the animal first inject cyanide and next inject sodium sulfate. Sodium sulfate can treat cyanide poisoning. So, animal B first happen ketogenous hypoxia and the next treatment. So the animal maybe survive, didn't die. Now, announcement. Don't let cyanide touch body because it is very pathogenic compound. Now you watch the experiment. Now cyanide, potassium cyanide. Now first the injection. Inject potassium cyanide into the abdominal cavity. Now you observe the manifestation. Now the animal first inject potassium cyanide, and next inject normal saline. Now mature mouse B. First inject potassium cyanide, and next inject sodium sulfate. Sulfate can treat cyanide poisoning. So the animal first happen ketogenous hypoxia, and next treatment. It is animal B. It is animal A. You observed animal A respiration decrease. Now collapse. Now animal A die. Animal B survive. Now results. Now potassium cyanide, mouse A and mouse B, normal respiration and normal color. Now after hypoxia, mouse A respiration 24/13 and even stop. Now color. Animal A rosy, light color of rose. Now survive 2 minutes 40 seconds. Now animal B, because first happened ketogenous hypoxia and the next treatment, so it didn't die, survive. Analysis and summary. Also have 4 questions. Which types hypoxia caused by cyanide poisoning belong to? Now you know belongs ketogenous hypoxia. Now what's the color? Rosy. Now also called the pink red flash. What's the pathogenesis of cyanide poisoning? Now why sodium sulfate can treat cyanide poisoning? Now you know it belongs to ketogenous hypoxia. Color of skin, ketogenous hypoxia is rosy, also called the pink red flash, because the oxygen content in capillary blood increase. Why oxygen in the tissue capillary blood increase? Now, what's the pathogenesis of cyanide? Now, the pathogenesis, cyanide can inhibit of mitochondrial oxidative phosphorylation, so ADP is decreased and lead to cell damage. Now, because utilize oxygen decrease, so tissue cell take in oxygen decrease, so in tissue capillary blood oxygen will more, so the color is rosy, like arterial blood. Now why can sodium sulfate can treat cyanide poisoning? Sodium sulfate can treat for cyanide poisoning because cyanide can react with sodium sulfate and form non-toxic compound, sodium cyanide. Sodium cyanide is corrected by kidney. Now the next you observe the color of liver. Now it is the first hypotonic hypoxia. Color of liver is bluish. Now the second carbon monoxide poisoning. Color of liver is cherry red. Now cherry red. Now the third is nitrate poisoning. Because from ferric state, so the color is coffee. Now the last, cyanide poisoning. Because utilized oxygen decrease, so in the tissue capillary blood oxygen more. So color is rosy, pink-red flash. Now it is a four color. Different hypoxia, color of skin different. Now, section 6, conclusion. Now the first, respiration in hypotonic hypoxia first increase and the next decrease, even stop. While respiration in other types of hypoxia decrease all the time, no increase. Now the second, tolerance ability for hypoxia in newborn mouse is stronger than that in mature mouse. Now the last, methylene blue can treat nitrate poisoning. Sodium sulfate can treat cyanide poisoning. Inhaling oxygen can treat carbon monoxide poisoning. Today, the hypoxia experiment is over. Now, thank you. Bye-bye.